This is the second part of looking at how genes are regulated in eukaryotes. In the first part, we looked at how the DNA is wrapped and how the accessibility of the DNA affects whether genes can be transcribed or not. In this second section, we're going to specifically talk more about the regulatory sequences found around genes in eukaryotes, and we'll look as an example at the Gall system in yeast. At the end of this, then, we're going to come back full circle to see how the transcription factors that are functioning, binding to enhancers near the gene, come back to regulate not just recruitment of RNA polymerase and the basal transcription machinery, but also how they can regulate the chromatin environment, or how tightly that region of the DNA is packaged. So just to review, eukaryotic transcription is regulated by transcription factors. And we have this region called the basal transcriptional apparatus. This is what is required for all transcription to occur. But that's really not enough all by itself for transcription to occur at high levels. In addition to the basal transcription apparatus, we also need other binding sites for transcription factors. And transcription factors do two main things. They can help to both bind and to stabilize this apparatus that binds at the core promoter, and it will also help to modify the chromatin environment in the area to make it more accessible for the basal transcriptional apparatus to bind. So, most genes have many transcription factor binding sites that all cooperate to regulate a gene. And some transcription factors, in fact, will bind to the regulatory elements of many different genes. And this is the way that separate genes, by requiring the very same transcription factors, can achieve coordinated gene expression, even though they are not found as operons, as is found in prokaryotes. So here is the regulatory promoter that's shown. So remember, the regulatory promoter is defined as being just within a few hundred bases of the core promoter. And then further away, there would be enhancer sequences to which other transcription factors would bind. Within the regulatory promoter, there are a number of binding sites for transcription factors that are commonly used among different proteins. For example, this GC box binds a certain transcription factor that is used in several different genes, whereas the OCT box or the CAT box are used by other genes. So for the rest of this video, we're going to focus on one particular example, which is found in yeast, and this is called the Gall system. Now, this is meant to be similar to the lac operon in bacteria, because Gall stands for galactose, and so galactose is an energy source that can be used by yeast cells. And if galactose is going to be used for energy in glycolysis, it has to be converted first into glucose 1-phosphate. And there are a number of synthesis steps that are required for galactose then to be converted into something that the cell can use. There are 4 different genes that are required, and these are the Gal1, Gal2, Gal7, and Gal10 genes. Gal7 is actually used in 2 different conversion steps, and these genes are only going to be activated if galactose is present in the cell and if it's needed to be broken down. So the way this works is the following. Each of these genes, these structural genes as we call them, these Gall genes are actually completely separate genes. Turns out that 3 of them are found on the same chromosome in a similar region of chromosome 2. The 4th one is found on a completely separate chromosome. So each of these Gall genes has its own core promoter where transcription will begin, and expression is regulated by a particular enhancer sequence called a UAS sequence. So a UAS sequence is an enhancer sequence that binds to a transcription factor called Gal4. Gal4 will activate transcription of the nearby Gall gene. So for example, these particular UAS sequences, if Gal4 binds there, then it will activate the Gal7 gene, whereas in order to activate the Gal10 gene, Gal4 needs to bind to these enhancer sequences near to the Gal10 gene. So now, instead of looking at all four genes separately, all four of those structural Gall genes, we're going to focus in on one of them. So Gal1 is the one we're going to look at. All four of those genes are regulated in the very same way. So this particular gene has to have a way to respond to the sugar galactose in the environment. And the way it responds to galactose is rather indirect. So Gal4 is the transcriptional activator protein that actually binds to the enhancer, but Gal4 is not the protein that actually also detects the presence of galactose. So there's another protein that does that, and we'll get to that in a minute. First, I'd like to talk about the situation where no galactose is present. So in this case, you do not want to activate those Gall genes because you have no galactose to break down. So if this is the case, what's going to happen is that Gal4 transcriptional activator protein will still bind to the enhancer sequence near each of the Gall genes. However, the Gal80 protein will bind to Gal4 and inhibit its function. Okay, so as long as Gal80 is bound here, it's masking the transcriptional activation function of that Gal4, and you would not get activation of the nearby Gal1 gene. In contrast, when you add galactose, there is this protein called Gal3, which will bind to galactose and change its conformation. When Gal3 changes its conformation, it gets a very high affinity for that Gal80 repressor, and it's going to pull that Gal80 off of the Gal4 protein. Okay, so increased affinity here means that now Gal4 is available to act as a transcriptional activator on the nearby Gall gene. And so when galactose is present, you turn on those Gall genes which are required for breaking down the galactose sugar. Next, I'd like to talk a little bit about what the Gal4 protein is actually doing to activate transcription of the nearby Gall genes. So that Gal4 protein is binding to that UAS sequence, and this insists on the nearby Gall gene by doing a couple of different things. So the first thing it does is Gal4 actually recruits a histone acetyltransferase, and this will acetylate all of the histones in this region of the genome. Okay, it will also recruit a chromatin remodeling complex. This complex is called SAGA, and move those nucleosomes out of the way so that the core promoter region will become accessible to the core transcriptional machinery. The next thing Gal4 does is it binds directly to the mediator protein, and the mediator protein assists in helping RNA polymerase and the rest of the basal transcriptional machinery to bind to the core promoter. The last thing that Gal4 does is it binds directly to TFIID, which is a component of the basal transcriptional machinery, and helps again to get that basal transcriptional apparatus bound onto the core promoter. So all of these functions of Gal4 result in the transcriptional activation of the nearby structural Gall gene. So Gal4 has this kind of two-step thing. First, it affects the chromatin environment, the nucleosome situation in that general area of the chromosome. And second, then it helps the core transcriptional machinery to get bound. There's a second regulation system that is also in play here, and this is very similar to what we found with the lac operon in bacteria. So just like the lac operon, so yeast cells would also prefer to use glucose rather than galactose. So glucose can go directly into glycolysis, doesn't need these conversion steps. So the way this works is that there is a protein that accumulates when glucose is high, and this protein is called Mig1. Mig1 is high when glucose is high, and Mig1 binds to a completely separate enhancer in each of those four structural Gall genes. MIG1 binds to this enhancer sequence called the MIG1 site, and when it binds, it recruits a second protein called TUP1. TUP1 is a corepressor, and whenever TUP1 gets recruited to this region of the genome, the Gall1 gene is going to be repressed. The reason why it's repressed is because TUP1 is actually a histone deacetylase, and if you recall, HDACs remove the acetyl groups from nucleosomes in the region and make that gene region less accessible. So all 4 of the Gall structural genes contain this MIG1 enhancer sequence, and if TUP1 is recruited to this region, it's going to actually act dominantly over the transactivation function of the Gall4 protein. So just to summarize this, just to compare and contrast prokaryotes versus eukaryotes. In prokaryotes, activators really do one thing, and they help to recruit RNA polymerase to bind to the promoter. Our example in the lac operon was CAP and CAMP, and it just helps RNA polymerase to get recruited more rapidly and effectively to the promoter. Okay, and in bacteria, if you have a repressor protein, it physically blocks RNA polymerase from binding. It physically binds to the start of transcription to inhibit transcription from occurring. Eukaryotes are a lot more complicated. Generally speaking, repressors make the nucleosomes wrap the DNA more tightly. So it makes that gene region inaccessible to factors binding to it and for the general transcriptional machinery to bind to the core promoter. Transcriptional activators can do a couple of different things. They can promote an open chromatin conformation so that the gene is more accessible. They can remodel the chromosomes, move them around, and to move them out of the way. And they can also help to recruit and to stabilize the basal transcriptional apparatus.